Okay, so let's go. So right now we are at the point where we have done the spinal cord, we have done the brain stem, we are skipping over the cerebellum until tomorrow morning, and now we're going to move up into the diencephalon and the cerebral hemispheres. And look, there's some really basic information I'm going to give you. You're going to look at the schematics and maybe think to yourself, do I have to know all that? Absolutely not. Just to iterate, so cerebellum will be tomorrow, and now it's we're ascending and we're heading to the diencephalon and the telencephalon. So I had mentioned the fact that the diencephalon is composed of the thalamus and the hypothalamus. The hypothalamus is the territory of the endocrinologists. So what about the thalamus? So look, the only thing you really have to know about the thalamus are maybe a couple of nuclei by their name. And the fact that what does it generally do? It's a sensory relay center. Now, the two major pathways we've done so far, right? Posterior columns, medial lemniscus, and anterolateral. What was it doing? It was relaying sensory information from the body to the cortex. And that's what it was doing. And that's, in fact, what the thalamus does. So the hypothalamus, as it says there, endocrine and autonomic for another time. The thalamus is a deep structure receiving all sorts of information from a lot of different places, and we'll definitely show you that again tomorrow with regard to both the basal ganglia and the cerebellum. But look at that thalamus projecting onto the cortex, right? Sending all those major projections to the cortex. And that's really what it's doing. If you think it's relaying information from where to where, in the case of the sensory modalities, right? From the entire body, the head and the trunk, right? It's relaying it from the body to the cortex, where you then project it onto the homunculus and you have that conscious awareness of where the information is coming from. Now, there's one little concept I'd like to just get across to you, and this is this idea of projections. So if you look at just the direction of these arrows, some of these we've done, some of them we haven't done. So reticular formation, just mention that as a group of neurons found in the brainstem. Rostral and caudal reticular formation. What have we done here so far? We've done somatosensory. Those are our general proprioceptive pain informations, right? And they're all funneling into the thalamus. So you get this idea of I talk to you, but that's it. So the reticular formation sends projections to the thalamus, but that's it. So does the cerebellum, the basal ganglia, we're going to do tomorrow morning, and limbic system I'm going to mention now briefly. When we think about these sensory modalities, look at them. Auditory, vision, vestibular. So those are our special senses, are being relayed actually through the thalamus. And look at the one exception, olfactory. It's clearly very different from everybody else. Now, when you look at the thalamus and that central location it has, we can see the fact that the arrows are going only one direction for sensory in, reticular formation in, and then it projects out on. But look what's going on between the cortex and the thalamus. There's this idea of reciprocity, reciprocal projections. I talk to you, and you also talk to me. So that means there are neurons in the thalamus that project to the cortex, and cortical neurons that project back to the thalamus. And look at these two names. Remember I told you about how we formulate these names, these proper names in neurosciences. Where does the axon originate? Where does it terminate? Thalamocortical means it starts in the thalamus and projects to the cortex. Corticothalamic means it starts in the cortex and projects down to the thalamus. And that's how we can distinguish these reciprocal projections. Thalamocortico-corticothalamic. I'm not going to ask you about all this. The idea here is that you get the concept that the thalamus is receiving a whole bunch of information. A lot of sensory information. It's going to regulate motor information, right? So that's what we got here, basal ganglia, cerebellum, motor. Limbic is about emotion. Reticular formation is about consciousness. A huge relay center, bringing in information and then sending it to the cortex where it actually is all assimilated and properly worked through. Everybody but olfaction. Now, this is one of those schematics that you look at and go, oh, good grief. You know, I have to know all this stuff? Absolutely not. We've only done, so far, 2 of all of these nuclei here for the thalamus. And I told you there are many nuclei. But the only ones you're really responsible for so far is this VPL, which is receiving all the information from the contralateral body, the neck down, and the VPM from the contralateral face, right? So information, sensory information being funneled in. But different parts of the thalamus are doing different things. We've got basal ganglia, we've got globus pallidus, we've got all sorts of crazy things going on there. You don't need to know these nuclei, you don't need to pick them out, you don't need to figure this out at all, okay? It's just the fact that we've got this inflow of sensory information that's being processed. And that's it. That's what you have to know about the thalamus. That it has this central location within the brain that is receiving inputs from a variety of different places. It relays sensory information, a lot of other information onto the cortex. So what about the cortex? It's straightforward anatomy. There's a frontal lobe, parietal lobe, temporal, and occipital lobe. And sometimes these lobes are distinguished from each other because of really deep sulci or grooves that you find. And we know that the human brain has these bumps or ridges, which we call the gyri, which is actually cramming so much of this cortical material inside of our cranial vault. The central sulcus is a deep, prominent groove that starts at the midline and passes all the way down laterally, right down to this very deep, deep groove, the Sylvian fissure. So that central sulcus, importantly, divides for us between the primary motor cortex anteriorly and the primary somatic cortex posteriorly, which means the fact that primary motor cortex is in the frontal lobe and the primary somatosensory cortex is in the parietal lobe. The frontal and parietal lobes are clearly separated from the temporal lobe here by this very deep Sylvian fissure. But where actually temporal ends and parietal begins, we kind of make up a line here. There's no deep sulcus whatsoever. So there's kind of like an imaginary division right about there. And then if we take a midline view, we can see the fact that that central sulcus does not continue into the midline structure here, but rather, again, we have to draw one of these imaginary lines down here. But again, we clearly have the frontal and the parietal lobe. And parietal is separated from occipital by this very prominent parietal occipital sulcus. And then we actually have the temporal lobe as well. So, 4 lobes. Sometimes there are fissures or sulci that separate them, and other times there are not. This large band of myelinated axons that connect the 2 hemispheres is the corpus callosum. And so to actually get this view, you need to take a scalpel and cut through the corpus callosum. And hopefully Dr. Coe has a break for us tomorrow afternoon. And we'll actually do that, and then you actually can turn the 2 hemispheres of the brain open like that to reveal this medial, mid-sagittal view. The corpus callosum has these different parts, the rostrum, the genu, the body, and the splenium. The genu refers to like a knee bending, the rostrum, the front part, and the splenium is behind. And within here, we actually have these large myelinated axons that are interhemispheric. They're connecting the left and the right cerebral hemispheres. Now, there's a lot of intrahemispheric connections going on as well. That is, frontal lobe talking to parietal lobe. Remember our arrows going back and forth between primary motor cortex, primary somatosensory cortex, talking on the same sign. But we also have lots of information going from left to right, right to left. And to do that, it all funnels through this huge structure here that's composed solely of myelinated axons, the corpus callosum. Then we take a top-down view. We can see our frontal lobe, parietal lobe. We can see that deep sulcus, the central sulcus, separating again frontal and parietal, primary motor, primary somatosensory. And then you have the sagittal fissure, or the interhemispheric fissure.
And if you were to hold the brain and just pry it open, when you look down there, you would actually see the corpus callosum. Turning the brain over now, we can see the frontal lobe, temporal lobe, occipital lobes, and a couple of structures that we know are important for a variety of different reasons, right? So I show you the olfactory bulb and the olfactory tract only to say, remember, this is not the first cranial nerve. This is the part of the brain that now begins to process olfactory information. We know that these structures here are incredibly important. Optic nerve, optic chiasm, optic tract. So that's two separate optic nerves are in front of the chiasm, and extending behind it is the optic tract. And remembering about the fact that there are nasty pituitary adenomas that can grow, we know that the pituitary attached to the hypothalamus has got to be right behind that chiasm. And the hypothalamus is actually going to be deep in that structure. How do I know that this cross-sectional view of the brainstem is at the level of the midbrain? What anatomical landmark did I mention earlier that we use to really clearly identify that this is the midbrain? Well, there are those two round structures, those red nuclei, that sort of give it away. And if you're really being on top of it and say, well, I also see those two big nerves, which I have a feeling, since they're so close to the midline, may in fact be ocular motor. And sure enough, that is actually ocular motor. But the midbrain is actually defined, can be defined by the presence of the two red nuclei. That little opening here is the cerebral aqueduct connecting the third and the fourth ventricles, and that gray matter around it is that periaqueductal gray helping in the processing of pain information. I've already gone through Brodmann's stuff. It's just out of interest that you know why that there are numbers associated with all these different parts of the cerebral hemispheres. So it's just based on histology, matching, function, which was very, very cool. So what do we have now? We've actually got a few things going on here in the cerebral hemisphere. So the cerebral hemispheres have got to be having a lot of things happening there. But so far in the course, the only two things we've done is the primary motor cortex, primary somatosensory cortex. And look how the two homunculi just match up beautifully, right? Trunk, arm, hand, face, right? And then we look more in the midline, we actually see what? The leg and the foot. And they line up so nicely. So it's not like you have to have, remember, two homunculi. It's just they're both identical. So there's that central sulcus with the primary motor cortex in front, primary somatosensory cortex behind it, frontal lobe versus parietal lobe. Now, there are a couple of other centers on here that we're going to talk about today, and then one more for tomorrow. And if you think about it, you know, there've got to be multiple different areas here in this brain, none of which we're going to get into. So we're going to keep it limited to a very few number of cortical areas that you actually are responsible for. Now, right here is motor about the face, right? How do we actually do motor? And one of the most important things about motor of the face is that ability to speak, right? So the fact that I am actually formulating words relies on these cortical bulbar neurons here to make sure that what? That the facial nucleus works, my pharyngeal and laryngeal muscles work, and my hypoglossal neurons work. And these are where the cortical bulbar neurons are originating from, this part of the cortex here for face. That's the mechanical side of it. But what about from my ability to actually know how to formulate words that you can understand? I mean, I can simply move my mouth, my tongue, etc., but it's the actual formulation of words that distinguishes us as human beings, right? And importantly, there's this area right next to the motor aspect of speech, which is really where you generate speech, and that's Broca's area. And Broca's area occurs in the dominant hemisphere, and for most of us being right-handed, that's in the left hemisphere. So this Broca's area about the formulation of speech, how do we make ourselves understood, is immediately adjacent to the motor centers for speech. Well, what a great place to put it, right? It should all be connected together. When you talk to your patient and you ask them a question, there's an important event going on, and that is that they understand what's being asked of them. You ask them a question, they have to understand it, and then they have to respond. And they have to respond in a way that you can understand them. The way they respond that you understand them is Broca's. And how they understand what you've said to them is another area in the superior aspect of the temporal lobe, which is Wernicke's. And if you can imagine for human advancement, for us being a human beings, this interplay between understanding speech and generating speech, right, is absolutely fundamental. And it shouldn't be at all surprising that these two areas are linked to each other, that they're actually talking to each other. And we're going to do that in just a moment. The other cortical tissue we're going to talk about tomorrow morning is the primary visual cortex that we see on this mid-sagittal view in the occipital lobe. And we'll talk more about that tomorrow with regard to visual cortex. So there are a lot of these places in the brain that have a singular function. There are a lot of places in the brain we actually think of them having a primary, a single function, or actually many different things going on together. And so there are very few of these primary areas. We actually have a primary motor, primary somatosensory, and we have a primary auditory. And look where primary auditory on that superior aspect of the temporal lobe is located, right next to Wernicke's. It's one thing I hear something. I can hear someone speak to me in Russian, but I don't understand it. I hear it. That's primary auditory. My understanding of the language is Wernicke's. So you're understanding me because we all speak English. You hear me speak and you understand what I said. And so it makes sense again that you put these two areas side by side with each other, and they happen to occur in the superior aspect of the temporal lobe. If there's a primary auditory cortex, chances are there's a primary visual cortex, and we just did that. That's actually back here in the occipital lobe. And then you look at the brain, you think, my gosh, there's a lot of the other part of the brain that just seems to be helping out or has maybe one other function. So Broca's and Wernicke's I've mentioned specifically, but a lot of the other parts of the brain seem to have this heteromodal thing going on here. Right? Many things are happening in these big bits of tissue. Or we actually have supplementary or secondary parts of tissue. So from my view of neuroanatomy, from a clinical perspective, that you recognize primary motor cortex, primary somatosensory cortex. That you recognize Broca's area in the dominant hemisphere and Wernicke's. And that you recognize that there's a primary motor cortex, primary auditory cortex, and a primary visual cortex. What else do you need? Right? Because all these other things are going to lead to very mixed signs in a patient that, will definitely cause, a lot of consultation and trying to figure out what's wrong with that individual. So, we know that stroke is a major disease state in our society. And so it absolutely is imperative that you have a basic knowledge of these vascular territories of the brain. We did a few this morning about the brain stem, and now we need to do a few with regard to the cerebral hemispheres. And without a doubt, the one artery that provides the lion's share of vascular blood to the cerebral hemispheres is that middle cerebral artery. And look at its huge territory. And if you look at it, look at the important things now that I've identified that are here in that middle cerebral territory. We've got the hand and the face, or primary motor cortex. We've got the hand and the face for primary somatosensory cortex. We've got Broca's area for the formulation of language. We've got Wernicke's area for the understanding of language. And we've got our primary auditory cortex for receiving sound. Lots of important bits of information being processed there in the vascular territory supplied by middle cerebral. Anterior cerebral certainly is important, but it's really doing more for that leg and the foot, and probably from the hip, the leg, and the foot for actually, when you think about the homunculus, for both the primary motor cortex and the primary somatosensory cortex. And then when it comes to posterior cerebral, there's one area of the brain that relies on that fundamentally, and that's going to be that primary visual cortex, which again we'll do tomorrow morning. So these middle cerebral artery, and this came up the other day, so I just want to iterate probably something that Dr. Cooper's already done. This middle cerebral artery is a major branch off the internal carotid. And it immediately goes deep into the brain, giving off these lenticulostriate arteries, which supply the basal ganglia tomorrow morning. And then it comes out superficially, and it either goes superior or inferior over what? Superior over the frontal and parietal lobe, and inferiorly over the temporal lobe, to provide that huge area of vascular supply that was on the chart before. And then we can see how posterior cerebral comes back here specifically for visual cortex, and anterior cerebral for the more medial aspects of the primary motor and the primary somatosensory cortices. So we've already done this. This is just more of the same. Now, we'll do that tomorrow morning because I want to let you get out of here early. So I give you these only for the fact that I'd like for you to look at them, and there's no expectation on my part that I'm going to ask you very specific questions about these very deep, very specific vascular territories. But the fact you get a general idea that the middle cerebral artery is a major supplier of blood with its superficial superior and superficial inferior branches to the frontal, parietal, and temporal lobe. And when we think about the frontal lobe, you're thinking about the face and hand primary motor, face and hand primary somatosensory, and Broca's on the frontal lobe and Wernicke's on that temporal lobe. So really important artery for those 4 areas of cortical tissue. Okay. So if we just now go, because it's all about cortex, what did I do there? What else, I guess? And I'm just going to introduce you basically, no, not that one. Should be another one. Higher cortical. Okay. So it's perfect to actually put these two side by side because we've kind of done all that already. So that's just iteration. That's iteration. Right? So you can read this for your own interest. But the important thing here is about this hemispheric localization. And what we're really thinking about is this idea of Broca's area being in the dominant hemisphere. And for the population, 90% of us are right-handed, which means that dominant is the left hemisphere. So you can read these. There's no expectation on my part for you to have any in-depth understanding about this. This is certainly stuff that you would come back to with Dr. Cooper in 5th term. What he would talk to you about, certain brain lesions or false localization, disconnects, etc. So here we actually have our 2 areas. Now I know unfortunately they have those numbers associated with them again. I provide you with those because I think that's important, because I know people may use that in the future. But as long as you have this very specific idea of what these 2 areas are doing, and the fact there's this preference for dominant hemispheric localization. So we actually have Broca's area here in the frontal lobe, the inferior aspect of the frontal lobe.
and we have Wernicke's over here in the superior aspect of the temporal lobe. And Wernicke's, by being adjacent to the primary auditory cortex, it makes perfect sense that it has everything to do with the identification and comprehension of meaningful words. Right? I get it. I know what's being said to me. And that Broca's is all about your formulation of words, that I make myself understood in the language that we share together. And you know that's definitely important, right? Because if any of you have started to try to learn a second language as an adult, right, you probably can't get your mouth right around those words, right? And that's a big deal. Because to make yourself understood in this new language, right, you really need to start training your mouth, your tongue, your pharynx, and your larynx to make sounds that are understood in that language. And so it's not merely just churning out sound, it's churning out meaningful, comprehensible words that are understood by the native speakers. And so we have these two very important areas. And it makes just intuitive sense that they would be connected to each other. And there are these arcuate fibers, these arcuate fasciculations that connect, that go back and forth between these two areas. Because I need to understand what's being said, and I need to respond. You can read about this. It's just basically more of the same, but there's a lot of information here about all those numbers, so don't get bogged down in that. And then vascular territory, I can't emphasize enough how we really think about middle cerebral. So if you have a very tiny lesion of middle cerebral, remember our motor lesion, a little tiny one, you'll just maybe knock out what? The hand or the face. And if it just knocks out the hand or the face from a motor point of view, that's bad enough. But if the lesion's just a bit more extensive, right, it may creep into Broca's area. But a massive stroke of the middle cerebral artery could affect Broca's, Wernicke's, you have motor, somatosensory deficits, right? So clearly, middle cerebral on the dominant hemisphere is actually very, very important. I'm only going to mention this because it's worthwhile that you have some idea that when we think about Alzheimer's disease, it really is a disease first and foremost of cortical tissue. And that's not to say that the brainstem and cerebellum don't have degrees of pathology associated with them, but when you think about neurodegenerative diseases, right, and where they predominantly localize, it's Alzheimer's is predominantly found, the pathology rather, is predominantly found in the cerebral hemispheres. And this occurs in the form of these things called senile plaques, neurofibrillary tangles. You get gliosis, which is the activation of glial cells, neuronal loss, shrinkage, etc. And so the cerebral hemispheres are profoundly affected in Alzheimer's disease. And this is just showing you relative distribution. And we know that if you think that Alzheimer's disease is characterized by what? An inability to learn new things and a loss of memory, then it would make sense that one of the most affected areas would be this little structure down here, which I'm just going to mention now, and that actually is the hippocampus. So, cerebral hemispheres, there's a lot there. Don't need to know it all. Broca's and Wernicke's area, yes. Where are they found? Yes. Who's the artery that supplies them? Yes. Is there a dominance feature to them? Yes. All right? And don't worry about all these other areas of the brain, these wonderful colored schematics like this, where we haven't done things. Don't worry about them. Okay? So, see, I'm really chugging along here. Right? So now limbic. There's only a couple of things I want to say about limbic. And I only do limbic because I'm always concerned that psychiatrists are going to come at some point in time and say, you've had your neuroanatomy of the limbic nervous system. You're going to say, I don't even know what it means. Now you'll be able to say, well, I remember the word limbic, but he told us that there was too much we needed to know. And look at this crazy schematic. I mean, I cannot expect you to know all this. I don't even know all that. But if you think that the limbic nervous system is all about emotion, and that there are other aspects of emotion and drive, memory, olfaction, and homeostasis. And what part of the brain would you expect to do that? Find that thing. So if I tell you limbic and olfaction go hand in hand, you can get that. So the olfactory nervous system is cranial nerve number 1. Memory, one word in the brain goes with the word memory. Hippocampus. The hippocampus is where we store memory. Emotions and drive, it's the amygdala. And certainly there are going to be psychiatric conditions you will come across where drives are absolutely out of kilter, and it probably has a lot to do with the amygdala. And then the hypothalamus, which is all that endocrine stuff, is I'm not going to be doing. So what do I really want to tell you? So there's a little mnemonic, home, about the limbic nervous system. So I don't want you to find the limbic nervous system on a brain. I mean, you can see that it's pretty widespread. I don't want you to have to learn all these cortical bits of tissue. Just the fact that you recognize that corpus callosum, yeah, you remember what that is, and that there's a lot of cortical tissue around the corpus callosum. That's about the limbic nervous system. That's where all this stuff is being processed. What stuff? Olfaction, learning, memory, right? So when it actually comes to these things, we give you an example. So smell conjures up can conjure up tremendous emotions and memories very quickly. So I know at some point in your life you've all walked into a bakery or you've walked into some environment where there's a wonderful smell, and perhaps for that split second, you're a child again, in your grandmother's kitchen, she's making something, and you have a very pleasant memory of something. I mean, that's incredible, right? So that olfaction has triggered something in your memory. And you can see then how these two aspects of your existence are tightly linked. Olfaction and memory. Certainly we see things, we hear things, we touch things, absolutely. But this is a very primitive part of the brain. And so this is something that is really shared by many vertebrate species. This very tight link between olfaction and memory. So if you think that you're a predator or you're prey, if you're prey, the smell of a predator is bad, and if you're a predator, the smell of prey is really good, right? And it links very important memories in your brain. The word hippocampus comes from the Greek word for seahorse, and I guess for some early anatomists, the hippocampus kind of reminded them of a little seahorse. I don't know. If you remember the term hippocampus as memory, that's fantastic. The CA fields, again, early anatomists had some creative ideas that the different parts of it reminded them the horn of Ammon, that ancient Egyptian ram-headed god. I don't see all this stuff. The average neuroanatomist, forgets what CA fields mean anymore. They just know it's CA1 versus CA4. But having said that, though, it's a very small, deep little structure, this hippocampus. And yet, you think about it, it couldn't be more important for you. Because this afternoon, when you do your quiz, you're going to be relying on your hippocampus, right, to recall the information you learned to actually put down the correct answer for the quiz. And if you think about your short-term and long-term memory of things, so how do we actually get around to doing this? Well, for the really important things in life, like your 12 cranial nerves that you're always going to be using, like this afternoon and with me, right? My quiz and the quiz this afternoon, that has got to be committed to long-term memory. And there's this process in the central nervous system called long-term potentiation. And this is a process by which synapses are modified to hold on to that information for a long time. Because there's lots of information we need for a very long time. And your cranial nerves is one of them. Your name, birth dates, special events. But you may be able to remember what you had for lunch yesterday. Maybe a real challenge to start thinking exactly what you had for lunch the day before. I'm trying to get a scenario where you have a different lunch every day, right? But, you know, last Tuesday, what did you have for lunch? It's not important. You know you had lunch. You probably vaguely remember having lunch with friends and a particular topic being discussed. But the exact intricacies of lunch, not all that important. And the brain is really good at figuring out what you need to keep and what you need to get rid of. 12 cranial nerves you've got to keep. What you exactly had for lunch last week, not so important. And it's this idea of modifying synapses, strengthening synapses, that allow us to go through life and retain this. And this event, this real cellular chemical event occurs in the hippocampus in this process called long-term potentiation. So I want you to be familiar just with the term in case it ever comes up with psychiatry. You can read these things for interest, and the amygdala with regard to drives, it's just straightforward. But again, you know that when I put these things up here, I'm not expecting you to tell me the pathways of the amygdala, where to find the amygdala, none of that stuff, right? If you just remember emotions and drives, like sexual drives in human beings and in mammalian species resides in the amygdala, that's absolutely fantastic. Gesundheit. So the limbic system is something that's going to come back to psychiatry. I mean, they're going to talk to you about because of emotions and drives, etc. You're going to probably get the limbic system when you have the gerontologists come in talking about Alzheimer's disease and the hippocampus, right? And maybe even a bit about olfaction. As long as you recognize that this is a part of the nervous system that's large, diffuse, and as we like to think, a bit ancient. And if you just remember that easy mnemonic, home. So for homeostasis, right? Olfaction, memory, and emotions. That sums up the limbic system. And just to associate the word memory with hippocampus as a place in the brain where you store your memory, and that it's not surprising then when you look at the hippocampus of someone who's died of what you suspect to be Alzheimer's disease, and you find there's a 50% loss of neurons in there. No wonder they were unable to recall important events in their life or important names of individuals, family members, right? Because that's where all that information is stored, and if you lose those neurons, then that's why you end up with something like Alzheimer's disease. All right, so I went a little over in time, and I know it was a little fast, but it's when you go back and